Hello, I'm Hanz Benedict Aguilar, a BS Biology student from the University of the Philippines Los Baños, UPLB. The behavioral study that I am presenting is part of my undergraduate research project supervised by Mr. Emmanuel Rafael from the Avilon Wildlife Conservation Foundation and Dr. Leticia Afuang from the Animal Biology Division, UPLB. This study is entitled Behavioral Characterization and Ex-situ Time Activity Budget of the Siebenrockiella leytensis Philippine Forest Turtle in Avilon Zoo, Rodriguez Rizal, Philippines. One of the functions of zoos is to conduct educational awareness and research to advance our understanding of threatened species, which are key subjects of rigorous studies on their behavior. In captivity, current studies prove the use of ethology in determining certain behaviors that can be considered as novel information. However, it is common that reptilian ethology in captivity is less likely to gain attention compared to mammalian and avian ethological research. Philippine Forest Turtle Siebenrockiella leytensis is critically endangered as listed in the IUCN Red List of Threatened Species. It is a member of the ancient family Gyrimididae and is the only endemic forest turtle from the Dumaran and Palawan Islands, Philippines. Studies showed that it is one of the understudied species and most secretive Southeast Asian colonial. Thus, unfolding its secretive nature is necessary to further understand its natural history as it is being understudied. Now, in line with this, the study aims to construct a preliminary histogram, describe general behavior pattern, record time activity budget. The study was conducted at Avilon Zoo within the 7-day observation time period. Data collection was done by direct early observations coupled by opportunistic video recordings using my iPad and smartphone. Turtles were directly observed and recorded for its daily activity from 6 a.m. to 10 p.m. time frame within 7 consecutive days. 5 minutes were allotted for each early instantaneous scan sampling observation to record the behavioral states of the 46 turtles. Video recordings were used to capture and verify both the regular and specific active and opportunistic behaviors. An ethogram was constructed based on the initial observation period, which was done by describing the movements of each behavioral states. The study obtained novel findings of behavioral data from 46 captive turtles within the week-long data collection. Preliminary observations revealed behavioral repertoire of the captive turtles consisting of the affiliative, feeding and foraging, swimming, walking, no locomotion or resting, and on-land states. These behaviors composed the ethogram together with their descriptions shown in this table. All of the states except the on-land occurred underwater. Results showed that the majority of the turtles were on land but not buried. The daily underwater activity peaked during day 4. The mean frequency of turtles on land decreased, and the movement of turtles from land to water increased. The portion of time observed underwater had a variety of active behavioral states, as shown in this figure. Daily allocation of time, also called time activity budget, between 6 a.m. to 9 a.m. time, has high frequency of activity occurrence followed by state of inactivity at midday starting from 10 a.m. to 5 p.m. Afterwards, from 5 p.m. to 10 p.m., the turtles return to their variety of active states showing behavioral variations, mainly underwater, as shown in this figure. Cumulatively, the turtles spent most of their time resting either on land or on water during daylight, suggesting that they may be saving their energy in preparation for their active states during the night. Efficient energy allocation correlates to the turtle's distinct behavioral and physiological traits. As an ectotherm, these freshwater turtles rely on environmental heat sources, and thermoregulation of this species mainly involves the use of water, owing for their aquatic nature. Increasing frequency of activity together with the increasing number of individuals performing the underwater behavior were all observed starting from 4 p.m. as the day transitions the night up to 10 p.m. Unfortunately, behavioral information for the closely related Siebenrockiella species is noticeably lacking, which limits the possibility for more accurate comparison. This is, as far as we are aware, the first ethogram for the endemic turtle species of the Philippines. Furthermore, ethological investigations in zoo-based populations yield significant insights into their welfare. The study recommends further investigation on the turtle's behavior during 10 p.m. to 6 a.m. suggesting a complete 24-hour behavioral monitoring of the turtles. I would like to extend gratitude to the following organizations and institutions, namely the Department of Science and Technology, the Avilon Zoo, the Biodiversity Conservation Society of the Philippines, and the Animal Biology Division under the Institute of Biological Sciences of the UPLB for helping and guiding me to accomplish this research work. Thank you for listening.